Hey, it's Jay. I'm excited you're going to be coming to Open Classroom. I wanted to give you a quick orientation so you're going to get the most out of the visit and hopefully the class will flow smoothly with the students. So what I wanted to talk about is first off what you should expect when you come to the class, what the students will do for preparation, and give you a little bit of background information so that you'll be on solid ground when you come in. Now, as far as what to expect when you come into the classroom, when you come in, I'll hand you an iPad, and assuming I get some extra logins from the textbook manufacturer, you'll take an electronic quiz, and you don't have to get the answers correct. We start with an individual portion, which should take just a couple of minutes. And then the students get together in groups to discuss the answers, and the majority of the grade is on whether the group gets the answers correct. So I hope that you'll join a group, and I envision your role as a student or learner. And this gives other students opportunities to be experts to help explain some of the material, which is a valuable learning exercise. Now, after the Learning Catalytics quiz is over, we'll stop, and still in small groups, I'm going to throw out a question about material we haven't yet covered. And the question, you'll be able to solve the question with the principles that we brought to class, but it'll be a new situation, and there's no obligation to get the answers correct, but to toss around the ideas. And after that, hopefully we'll be finished with that in the first 15 minutes of class. We'll head back to individual desk, or a lot of times students will stay in their groups, and we'll go through the material from the chapter. So my hope is by giving you a little bit of background information, you'll be able to understand the conversation that I hope you'll have with the students. As far as student preparation goes for the class, we go off a couple of different websites. The first one is Moodle. You're familiar with this. We have our week in Moodle. And then for each class, we have an outline of what we're going to do. If you're coming to Tuesday's class, we'll be starting with the lymphatics, and we'll probably move on to the immune system. If you're in Thursday's class, we'll be looking at immunity. So I'll give you links to these documents. For the lymph class, what we're going to see is we have a single learning objective, which is explain the different causes of edema. It has the homework that's going to be due. This is what the quick quiz will be on. And then what we're going to go through. If you're coming to Thursday's class, we'll be talking about the immune system. And for the homework, we have not only the dynamic study modules, but a quick video from NPR. It's kind of cool, timely about the flu. Okay, what are dynamic study modules? So dynamic study modules are an interactive way of going through the book chapters. I'm in masteringamp.com. This is a companion site to the textbook. It has a lot of lab exercises, some virtual anatomy resources, a lot of different things that are useful, but one of the most useful, I think, are the dynamic study modules, which is a way to go through and read the chapter in an inquiry-based way. Some students will read the chapter first and go through these modules. A lot of students will not even read the chapter, but use this as a tool for going through the chapter. And how it works is there are a series of questions. And when I get this first question, I'm going to click I don't know, and I'll show you what happens. Eventually, we're going to answer 8 questions. And after we finish, it's going to take me to an explanation for that particular question. So I'm going to submit that. And then look at this next question. Small proteins released from tissue cells infected with viruses, as well as from activated lymphocytes and macrophages, are interferons. Yay, I got it right. So that's good. Now, what happens if I get this wrong? Well, it tells me right away I was wrong, and then I'll be able to go through these questions, and at the end, I'll show you how it leads me to those answers. Okay, so now I have finished this first 8-question series. I got 5 right, 1 I don't know, and 2 wrong. Now it's going to take me to learn in the chapter. And when I go and learn, it will tell me I was incorrect. It gives me the correct answer and takes me to what I need to know. Now that will happen for each of the incorrect questions. It will give me the correct answer and an explanation, sometimes with a figure and sometimes with words, but that part of the chapter. And where I don't know, it also will take me to an explanation. So it's a great way to get the significance of a portion of the chapter before you read it or while you're reading it. And it breaks the chapter into bite-sized pieces. I can see my progress up here as I move along. So the idea is that students have completed the dynamic study modules and they arrive at class ready to discuss the information. And they expect to answer a couple of questions on it before we dig into it. So if that's the case, you're probably going to want a little bit of background information. You don't have to be familiar with stuff, but at least hopefully you'll have a general idea of what we're going to try to accomplish in each class. So for Tuesday, we're going to start with lymph, and I think that I can explain relatively quickly what lymph is. The first thing we need to know is what is interstitial fluid. Interstitial fluid is the fluid between blood vessels and cells out in our tissue. So what we see here is a blood vessel, this tube carrying blood to these tissue cells. Okay, tissue cells right here. And between the vessel and the cells, we have interstitial fluid. What happens in our body is we have blood pressure, and this pressure moves blood through the circulatory system from the arteries to the veins. But just like a balloon, pressure that moves fluid in one direction is moving it in all directions. So it's pushing fluid out of the vessel. And this blood pressure in this case is pushing at 30 millimeters of mercury. Now, in our capillaries, all these small things can pass through the walls. Things like water, oxygen, glucose, and amino acids. Large things like plasma proteins aren't able to cross the wall of the capillary. What this means is that there are lots of proteins inside the blood vessel that we don't see out in the interstitial fluid. So there is a lot of water in the interstitial fluid compared to the plasma. What this means is the concentration of water is higher in the interstitial fluid than the plasma. So water wants to go from high concentration to low concentration into the blood vessel. Nature doesn't like it when concentration of something is unequal, and it will try to equalize it. So we have two forces. Blood pressure is pushing fluid out of the vessel, and what's called osmotic pressure is pulling fluid into the vessel. It's osmotic pressure due to the proteins in the capillary. So the direction that fluid is going to flow is in the direction that the forces are strongest. I'm going to look here at the arterial end, where we look at the blood pressure. I'm just kind of highlighting, it's pretty dim. 30 millimeters of mercury at the arterial end, and only 15 millimeters of mercury at the venous end of this vessel. That's because as blood moves through the circulatory system, pressure drops, blood moving from higher pressure to low pressure. On the other hand, the concentration difference between the interstitial fluid and plasma doesn't change. It remains steady at 21 millimeters of mercury. So at the arterial end of this blood vessel, hydrostatic pressure is stronger than osmotic pressure, and fluid moves out of the capillaries, net movement out at the arterial end, and at the venous end, the hydrostatic pressure, blood pressure, is lower than osmotic pressure, so fluid moves back in. And we have a nice circulation from the plasma through the interstitial fluid. Well, it turns out that more fluid is pushed from the vessel into the interstitial space than is drawn back into the vessel on the venous end. What this means is if that fluid couldn't drain anywhere, we would have an accumulation of interstitial fluid or swelling. It's called edema. The way our bodies have gotten around this is we have these other capillaries here. They're called lymphatics. And as long as everything's working appropriately, any extra fluid that's not draining through or traveling out of the vessels on the venous end will enter the lymphatics and travel through the lymph system, through lymph nodes where it's filtered back into the venous circulation. So lymph is really a series of vessels that will carry interstitial fluid from the interstitial space back into the bloodstream, and that happens at a lot slower rate than in blood vessels. Whereas a red blood cell might travel through the entire circulatory system once a minute, their circulation of lymph would happen maybe a couple of times a day. Another thing I want to mention while we're in this slide is that the scale is off. When we're looking here, I'm just going to erase some of this stuff. When we're looking at the relative size of proteins and red blood cells, this protein looks like it's a similar size maybe to a red blood cell. Actually, the plasma proteins, the size is on the order of nanometers, whereas we look at the size of cells on the order of micrometers. What that means is the length of one of these proteins is maybe 1,000th of the size of a red blood cell, but this is in 3 dimensions. So a protein might be maybe 1/1000th of the size of a cell. They're much, much smaller. And even though they're too big to leak through the capillary walls, we need to think of these as much smaller proteins. That's because as we move on to the immune system, we're going to talk about antibodies, and antibodies are proteins which are much, much smaller than cells. Okay, hopefully you know now what to expect when you come to class, what the students have done for preparation, and have a little bit of background information on the lymphatic system. If you're coming for the immune system, I'll try to do another video later on, but obviously I'm running on. So look forward to seeing you in class.